Chelsea.
Hey.
So what are you showing us here today?
So this is Bio-Rad's I-12 host cell protein detection instrument. So what this does is it is a 2D process that allows you to detect any residual protein contaminants that are in your protein product, and also validate any host cell protein antibodies that are used for that detection.
Great. So can you walk me through the HCP detection workflow?
Sure. So the first step in the process is to actually do sample prep. So we have sample prep kits that allow you to remove any ions or detergents or lipids from your sample, allowing you to resolve the protein in its native state, allowing you to really thoroughly detect and have the best resolution possible. After you've done your sample prep, you then separate the proteins within the sample based on all of the protein's individual isoelectric points or PIs. This is done in these strips right here that have different pH ranges that are then placed in these channels that allow different currents to run across the strips. So you can have up to 12 different samples, or one sample with 12 different optimization components run within it. The good thing about Bio-Rad system is the fact that each channel or each current is run individually, so one poor sample preparation does not ruin or is not detrimental to any of the other 11 samples that are being run. So you can take and actually optimize with this system and then continually run multiple different samples. And if for some reason the sample prep wasn't done well, it doesn't have a detrimental effect on any other sample.
Great. So what happens after we've separated based on PI?
So after you've separated based on PI, you then separate dependent on size. So you would take that strip and place it in one of our Criterion TGX gels or Criterion TGX stain-free gels. So these separate based on the size of the protein, and you simply place the strip at the top of the gel and then run it as a normal sds-page gel. The difference between the Criterion TGX and the TGX stain-free is the fact that the TGX stain-free gels have a chemical compound contained within the gel that allows you to label tryptophan residues in proteins. That allows you to instantly see, after using a Bio-Rad imager, how well the actual separation was performed and if you have good resolution on the gel. Because you don't want to continue through this workflow if you actually don't have good resolution and separation based on either the PI or the size.
It's a good time to check.
Yes, exactly. You want to have multiple checkpoints throughout this workflow, because if something goes awry in the middle of the workflow and you don't check it until the very end, you don't know what happened and when.
So what's this apparatus over here?
So this is Bio-Rad's dodecacell. It allows you to run up to 12 gels at one time, which is great because if you're running 12 different samples on your I-12 here, either 12 different samples or one sample with 12 different conditions, you can immediately transfer them to a gel and run them all at once. And because these gels are fast running, they are all done within 30 minutes.
Oh, that's great.
Another great thing about this is the fact that you don't have to fill out this entire thing to run 12 gels. If you only have 3 or 6 samples that you need to run, you can just use 3 or 6 slots.
Oh wow, that saves a lot of reagents, I'm sure.
Yes, it does. Saves lots of reagents as well as time and effort in terms of doing everything.
Great. So then what's the next step in the process?
So the next step after you've run the gel is the fact that you want to take it into a Bio-Rad imager and see whether or not you have good separation and resolution of your gel. This again is using the stain-free technology and covalently modifying the tryptophan residues of the proteins.
Okay, and so then do we transfer next?
Yes, transfer is next. Okay, so after you run the gel, the next thing you want to do is transfer it to a membrane. So most people still use the wet tank transfer, which can take up to overnight. So what Bio-Rad has developed is the trans-blot turbo, and what it allows you to do is transfer in 3 to 7 minutes. So you literally pop one of these guys out, make a sandwich using one of our transfer packs, transfer the gel to the membrane, and then because you've labeled your proteins with the stain-free gels, that is a covalent modification, so you can actually see how well the transfer happened onto the membrane, which is important because you don't want to continue on and use an expensive reagent like Cipro-Ruby to detect your host cell proteins if that transfer didn't happen appropriately.
So as you mentioned, another checkpoint in the process.
That is correct. This is another checkpoint in the process to make sure that everything is on track to being a successful host cell protein detection.
That's great. Well, thank you for walking me through the whole process.
You're
I
welcome.
can see that with the multiple checkpoints, it helps ensure customer success.
Yes, that's what Bio-Rad wants to do. We want to basically give the customer as much success as possible and give them checkpoints along the way so that they know where things went wrong.
Perfect. Thank you so much.
You're very welcome.